My name is Brandon Wirtz, and the second video I will be doing is on the origins of blood cell types and how to distinguish between them. So the first ones I'm going to talk about are the myeloid lineage, which are the first responders to infection. The first one is a granulocyte. They are the front lines of an attack during an immune response and are considered part of the innate immune system. Granulocytes are white blood cells that are classified as neutrophils, basophils, mast cells, or eosinophils based on the differences in their cellular morphology. All granulocytes have multi-lobed nuclei that make them visibly distinctive and easily distinguishable from lymphocytes, whose nuclei are round. The cytoplasm of all granulocytes is replete with granules that are released in response to contact with pathogens. These granules can have a variety of functions, such as damaging pathogens directly. Some regulate trafficking and the activity of other white blood cells, including lymphocytes, and some even contribute to the remodeling of tissues at the site of infection. The next one we're going to talk about are neutrophils. They constitute a majority of circulating leukocytes, anywhere from 50 to 70%, according to our book. There's much more of them than eosinophils and basophils and mast cells. After differentiation in the bone marrow, neutrophils are then released into the peripheral blood and circulate for 7 to 10 hours before migrating into the tissues, where they have a lifespan of only a few days. The number of circulating neutrophils increases significantly and more are recruited to tissues, partially in response to cues the bone marrow receives to produce and release more myeloid cells. Neutrophils are recruited to the site of infection in response to inflammatory molecules, such as chemokines. They are generated by innate cells, which can include other neutrophils that have engaged a pathogen. Once in tissues, neutrophils phagocytose or engulf bacteria and secrete a range of proteins that have antimicrobial effects and tissue remodeling potential. Neutrophils are the dominant first responders to an infection and the main cellular components of pus, which is just a little FYI. The next one we're going to talk about are basophils. They are non-phagocytic granules. They contain large granules filled with basophilic proteins. They are relatively rare in circulation. In response to the binding of circulating antibodies, basophils release the contents of their granules, which histamine is the best known one of them. That increases the blood vessel permeability and smooth muscle activity. Basophils are very critical in response to parasites, particularly worms, but also in areas where worm infections are less prevalent. Basophils may also secrete cytokines that modulate the adaptive immune response. Mast cells are released from the bone marrow into the blood as undifferentiated cells. They mature only after they leave the blood. Mast cells can be found in a wide variety of tissues, including the skin, connective tissues of other various organs, and mucosal epithelial tissue of the respiratory and digestive tracts. These cells have a large number of cytoplasmic granules that contain histamine and other substances. Mast cells can also play an important role in the development of allergies. Next are eosinophils, which, like neutrophils, are motile phagocytotic cells that can migrate from the blood into the tissue spaces. Their phagocytic role is significantly less important than a neutrophil, and they are believed to have their most important role in the defense against multicellular parasitic organisms. They can be found clustering around invading worms whose membranes are damaged by the activity of proteins released from the eosinophilic granules. Eosinophils may also secrete cytokines that regulate B and T cell lymphocytes, thereby influencing the adaptive immune response. Next, we're going to talk about monocytes. They can make up about 5 to 10% of all of our white blood cells and are a heterogeneous group of cells that migrate into the tissues and differentiate into a diverse array of tissue-resident phagocytic cells, such as macrophages and dendritic cells. Macrophages play several different roles. Some are long-term residents in tissues and play a role in regulating their repair and regeneration. Others participate in innate immune response and undergo a number of key changes when they are stimulated by encounters with pathogens or tissue damage. The dendritic cells are critical for the initiation of immune response and are acquired or have acquired their name basically because they resemble the dendrites of nerve cells. They're able to extend and retract, which allows them to increase the surface area available for browsing lymphocytes. They seem to arise from both the myeloid and the lymphoid lineages of the hematopoietic cells. Dendritic cells perform distinct functions of antigen capture in one location and antigen presentation in another. Outside of the lymph node, immature forms of the dendritic cells monitor the body for signs of invasion by pathogens and capture intruding foreign antigens. They process these antigens, and then they migrate to the lymph nodes where they present the antigen to naive T cells, initiating the adaptive immune response. Immature dendritic cells take on their cargo of antigens in three ways. They engulf it by phagocytosis, internalize it by receptor-mediated endocytosis, or imbibe it by pinocytosis. After activation, dendritic cells abandon residency in the peripheral tissues, enter the blood or lymphatic circulation, and then migrate to regions of the lymphoid organs where T cells reside and present the antigen. The next type of cells we're going to talk about are the lymphoid lineage of the adaptive immune response. The first ones we're going to talk about are lymphocytes. They are the principal players in our adaptive immune response. They can represent up to 40% of our circulating white blood cells and 99% of the cells in the lymph, according to our book. They can be divided into three categories: B lymphocytes, which are B cells; T lymphocytes, which are T cells; and natural killer cells, which are NK cells. Lymphocytes are relatively nondescriptive cells that are very difficult to distinguish in morphology. For example, T and B cells look very similar under a microscope. Let's see, B lymphocytes, they derive their name from where they mature, which is in the bone marrow for us and many other mammals. Mature B cells are definitively distinguished from other lymphocytes and all other cells by their synthesis and display of B cell receptor, which is a membrane-bound immunoglobulin molecule that binds to an antigen. B lymphocytes can also improve their ability to bind antigen through a process known as somatic hypermutation, and can generate antibodies and several different functional classes through a process known as class switching. Ultimately, these activated B cells can differentiate into effector cells known as plasma cells. These plasma cells do not divide, and although some are long-lived, many die within 1 to 2 weeks. T lymphocytes derive their name from where they mature, which is the thymus, and they express a unique antigen binding receptor called the T cell receptor. Unlike the membrane-bound antibodies on the B cells, which can recognize soluble or particulate antigen, T cell receptors only recognize processed pieces of antigen, mostly peptides, bound to cell membrane proteins called major histocompatibility complex molecules. These molecules are genetically diverse glycoproteins found on the cell membranes. The ability of the major histocompatibility complex molecules to form complexes with antigen allows cells to decorate their surface, exposing them to browsing T cells. Major histocompatibility complex comes in two different versions: class 1, which are expressed by nearly all nucleated cells of vertebrate species, and then class 2, which are expressed by professional antigen presenting cells and a few other cell types during inflammation. The natural killer cells, on the other hand, are lymphoid cells that are closely related to B and T cells. They do not express antigen-specific receptors and are considered part of the innate immune system. They are distinguished by the expression of a surface marker known as natural killer 1.1, as well as the presence of cytotoxic granules. Natural killer cells constitute anywhere from 5 to 10% of lymphocytes in human peripheral blood. They are very efficient cell killers and attack a variety of abnormal cells, including some tumor cells and other cells when they become infected with viruses or a virus. They distinguish cells that should be killed from normal cells by recognizing the absence of the major histocompatibility complex class 1, which is expressed by almost all normal cells, but is specifically downregulated by some tumors and in response to some viral infections. Natural killer cells express a variety of receptors for the major histocompatibility complex class 1 that, when engaged, it inhibits their ability to kill other cells. When natural killer cells encounter cells that have lost their MHC class 1, these receptors are no longer engaged and can no longer inhibit the potential cytotoxic tendencies of natural killer cells, which then releases its cytolytic granules, which kills the abnormal target cell. And that concludes the different types of, or the origin of different blood types, or blood cell types, in both the myeloid lineage and the lymphoid lineage.